I'm going to use a new bubble sheet. I'm trying something out. So it's basically, I mean, there's nothing different about how the exam's going to be set up compared to other times. It'll be longer, but aside from that, nothing different. But instead of having like the do-it-yourself answer sheet thing, I'm going to have you guys do a bubble thing, and I just want to give it a shot. So just in the interest of you not being panicked when you see it tomorrow, you can see it now. You just fill in. Yeah. Well, sometimes not. No more or less than before. Like, you know, it'll be. It will depend on the question. Like, if I can come up with 5 options, I usually put in 5 options. If I'm stumped after 4, I stop at 4. Sometimes I stop at 3. But anyway, just so you can see it, that's what we're going to be using tomorrow. And you don't need a student ID or anything. You just write your name on top, but it'll I don't know. Like I said, I want to check it out. One of the nice things about it is it'll tell me if there are particular questions that people are missing at a higher percentage. So I'll be able to identify particular questions in a way that I wasn't really able to do when doing it by hand, totally by hand. Anyway, that's really all I have to do. So yeah. There are definitely more multiple choice than before. There are roughly the same number of fill-in short answers as a typical exam. I honestly can't remember how many there are, but there are more than 50 multiple choice questions. But I do expect people are going to be like, I'm sort of timing it so that you're going to take something on the order of, it probably is going to be something on the order of around an hour and a half. But that means some people will be done in 45 minutes. Sorry, that'll go away in a sec. And other people will go the whole time, and that's fine. But you shouldn't be time crushed in any way. But you know, don't expect to be out in 20 minutes either, unless you really want to fly. And I don't suggest doing that. That's usually not a good thing. So what should we talk about? I'm happy to talk about any topics you wish. Yeah. Yeah. Sure. Oops. I thought it was just... There we go. Function of... Get there. Here we go. This one? Yeah. Okay, so function of intrafusible versus extrafusal fibers. Make sure this thing's working. Well, it seems to be. Okay. Basically the idea is that extrafusal fibers are all the other fibers that are in a muscle organ. So like all the standard typical muscle cells that we've ever talked about, those are all considered extrafusal fibers. But we call them extrafusal fibers in this context to differentiate them from intrafusal fibers. The intrafusal fibers are these guys. So like this is one muscle spindle in one muscle organ. All of this out here, these are all extrafusal fibers, but there are these 2 little muscle cells that are in here that are called intrafusal fibers. The thing about them is there's only 2 of them, so their ability to contribute any kind of like real force to the muscle is basically negligible because there's only 2 and they're muscle cells. They're not, they're nothing more than that. The thing about them though is that because they're inside the muscle spindle, they allow the muscle spindle to change its length. So it's not really contributing like substantial force that could ever really be measured, but it is allowing the muscle spindle to change its length. And the point of the next slide, which came out of a different book, what it's trying to show is that if you didn't have that feature, what would happen is that as your overall muscle contracts, so going from here to here, then what would happen is the muscle spindle, which is connected to your 2 bones on either end of the muscle, directly, it would end up basically losing all the tension that was previously placed on it. So there'd be a bunch of slack in these connective tissue elements that connect it to the 2 bones. So what the intrafusal fibers allow this thing to do is to shorten. And by shortening, they allow it to basically stay calibrated with the length of the muscle itself that it's embedded in. So as a result, there's always some tension on these connective tissue elements, which means that if there's any added stretch that's placed on this muscle, the muscle spindle will be kind of calibrated and primed to be able to receive it. If you didn't do this, then when the muscle gets short, then the muscle spindle would just have a lot of flop and slack in there, and it wouldn't be able to pick up any new tension placed on the muscle. So again, the short story is the intrafusal fibers allow the muscle spindle to change length to keep it calibrated with the muscle that it's embedded in. Sure, since it's the next one. How's it different from the spindle? Spindle reflex? Yeah. Okay, so the tendon organ, actually it was shown, the best picture actually of it is here. It's, I guess it's actually technically encapsulated, but it looks to me like it's kind of like a bunch of nerve endings that are embedded in the tendon. But there is some connective tissue stuff in here too. Basically, the idea is that this is also a stretchometer, but it is, I tend to think of it as more like a fail-safe, so that if tension starts to get really strongly, if you start to have an awful lot of tension being placed on this tendon, it will cause the Golgi tendon organ to start firing, and that initiates a reflex that basically causes this muscle to shut down, which will relieve the tension that's being placed on the reflex, or on the tendon. So in this picture where it actually talks about this, so what this is showing is that if you have tension being placed on the tendon organ, that'll send a signal up a sensory neuron, which will then interact with some interneurons, and what they will do is they will inhibit, so there's a negative sign there, they will inhibit the muscle, the motor neurons that are leading to the muscle that's associated with the tendon. So they're basically, if this thing gets stretched really severely, it will shut this muscle down and relieve that stretch. So you can sort of imagine if you, I don't know, you're jumping from on high and you're landing on your leg at and there's a lot of tension being placed on that tendon at that moment, what this reflex will do is actually it'll kind of shut down your quadriceps, which is, could potentially mean you're collapsing into the floor, but at least you won't tear off that tendon or cause any damage to the tendon. So, like on its basic level, I think that's a way to think of an important job of this tendon reflex, is that it will prevent you from causing severe damage to tendons, because whenever the tension gets really severe here, it'll shut down the muscle and relieve that tension. The other thing it does kind of do is that it sort of dampens down the amount of force that this tendon is experiencing. So even it doesn't have to only fire under like severe dangerous conditions where you're about to do damage to yourself. It'll fire under kind of normal amounts of tension that would be placed on the tendon, and that'll kind of slow down and deaden, reduce the amount of force that your muscle is generating. It might not shut the muscle down altogether, but it'll kind of moderate the muscle a little bit. And so the result is if I go and pick up something that's, like this is styrofoam, it's not crazy fragile, but I guess in theory I could do some damage if I tried to really crush this thing. But when I pick it up, I do not need to crush it, right? And if you pick up a paper cup full of a drink, you do not need to crush it, you just need to hold it. And so under those conditions, the tendon organ will help you to kind of modulate how much force you're applying. As you start to put tension on your tendon that are associated with those muscles, it'll kind of dampen down your muscle's force and prevent you from over contracting and over crushing the thing that you're trying to hold. So the thing that's different is if you look at the sort of the general structure of this reflex, if you stimulate the sensor, the reflex will cause this muscle to be inhibited and will relax in the tendon organ reflex. In the muscle spindle reflex, it was kind of the opposite. So here's a muscle spindle. If you stimulate the muscle spindle, that'll actually cause stimulation of this muscle that the tendon is the spindle is embedded in. So they kind of have the opposite effects. They're sort of competing with each other. Like I said, this one is more about if you set the muscle to a particular length, this reflex helps you to keep it at that length, even if there's unexpected force applied to your limbs. Whereas the tendon organ is more about kind of dampening down the force that you're generating and modulating it, keeping it from getting over the top. Certainly keeping it from damaging your tendons. What else can we talk about? Yeah. For norepinephrine parasympathetic. Yeah, so all the sympathetic division, I think with the exception of sweat glands, it releases norepinephrine onto its effector organs. But like we saw also with acetylcholine, there are several different kinds of norepinephrine receptors. And the kind of norepinephrine receptor, the specific kind that you have, basically determines the effect on the target organ. Because it's norepinephrine in every case. But what differs is the particular kind of receptor. The alpha receptors, we kind of generalize in your textbook. There's technically two different varieties of them. There's alpha 1 and alpha 2. You do not really have to know the difference between them. Most of the time, these guys are excitatory. And so I think if you go through and look at all these different examples, these are all effects where there are alpha receptors on effector organs that are receiving norepinephrine. And in almost all the cases, there's some kind of excitation that's happening. So we've got vasoconstriction of blood vessels to skin. And dilating of pupils means you have to kind of contract your iris muscles. And let's see, what's other good examples? Increasing clotting and stimulation of orgasm, stimulation later. So like most of these things are excitatory. I think the major exception, if you kind of are going through here, where it's not excitatory would be the effects on digestive organs, where digestive organs tend to have their activities inhibited. So like we have a decrease in motility and secretions from our digestive organs. That's what this one is basically telling us. So in these cases, these alpha receptors are more inhibitory, but in most other cases, alpha receptors tend to be excitatory. If you look at other effectors, other sympathetic division structures, so here's a whole new set of structures. These guys all have beta adrenergic receptors on them instead of alpha adrenergic receptors. There's actually 3 different kinds of these. We don't really have to know the difference. In most of the cases, beta adrenergic receptors will tend to inhibit the target organ. So we actually talked in the parasympathetic, or I'm sorry, in the sensory chapter about how inhibition and relaxation of your ciliary muscle causes your lens to kind of get stretched out, which causes you then to focus on distant objects. I'm going to skip this one for a second, but vasodilation. So here we are. This is vasodilation in the vessels that lead to skeletal muscles and deep coronary arteries. Here you're inhibiting the smooth muscle, which then causes those vessels to kind of get pushed outward by the pressure inside them, the blood pressure inside them. So you'll vasodilate by inhibiting the smooth muscle. Bronchodilation works the same way. You're inhibiting the smooth muscle in the bronchioles, which allows those vessels to open up, those structures, and so you get better airflow. Relaxation in the uterus. Again, these are all inhibitory effects. The main exception is in the heart. You have, I think these are actually the beta 2 receptors that are in there. They cause an increase in heart rate, an increase in contraction. So they are beta receptors, but that's really the major exception where you have some kind of an excitatory effect. In most cases, beta receptors are more having an inhibitory or relaxatory kind of an effect. So I don't want you to kill yourself over this stuff, but at the same time, like, you should have a pretty good idea of what type of adrenergic receptor, alpha or beta, are found on these different structures. I think in general, if you can remember the couple exceptions, like the main exceptions would be heart, which have beta receptors on them, even though it's excitatory on the heart. And then digestive system tends to have more inhibitory effects, even though usually alpha receptors are excitatory. So remember those, and then if you can remember the rest of them based on what they're doing, most of the time alpha receptors are causing excitation and beta adrenergic receptors are causing inhibition. I think that makes it fairly easy to learn them. Yeah, sorry. So sweat glands receive acetylcholine instead of norepinephrine, which is weird. It does say that. I used red text at some point here. Here it is. So sweat glands, so this is when we were talking like these kinds of diagrams. Sweat glands, the sympathetic division causes them to increase their secretion. That actually is stimulated by acetylcholine as opposed to norepinephrine. I think these are the eccrine sweat glands. So these are like your standard generic sweat gland. If you start to exercise, you need to increase your sweat production. and it's acetylcholine and not norepinephrine that's hitting them. That's the one place where acetylcholine is used by the sympathetic division to stimulate its effector. Normally, acetylcholine is parasympathetic division neurotransmitter. But this is the exception. The way it gets away with that is that the parasympathetic division does not talk to sweat glands. So it has no say. And so there's no parasympathetic division releasing acetylcholine, which is what makes it not confusing for these cells if the sympathetic division releases acetylcholine, because that's the only input they're getting. Yep. Good. Yep. Oh, I was past. Where did you go? Here they are. Okay. So the big picture here is that in all these cases, you have, because it's sympathetic division, you have signals coming out of the spinal cord, either in the thoracic region or the lumbar region. They're coming out of the lateral horn of gray matter. And these axons then travel in two stages. You've got a preganglionic axon that leaves the spinal cord and goes to some ganglion, where there's a synapse, which then gives rise to a neuron that produces a postganglionic axon that goes to the effector. So there's sort of two cells along the way, and we always form a synapse in some ganglion somewhere. In the first two, so the spinal nerve route and the sympathetic nerve route, the ganglion happens to be part of the sympathetic chain of ganglia, which exists right alongside the vertebral column. So actually in the prior slide it shows that. So here's the vertebral column. There's your spinal cord, kind of normally would have the arches sticking over them, so we wouldn't be able to see it, but that's been removed in this picture. And the idea is that you've got coming out of your spinal cord, this would be your ventral root of the spinal nerve, which would then go through this fuse with the dorsal root and become the spinal nerve, which would then give rise to this thing. This would be your anterior ramus. But you can see we have these connections between the anterior ramus of your spinal nerve, which normally is going to go off to its destination, and then we've got these two communicating rami. We've got a gray communicating ramus and a white communicating ramus. So the first route, this spinal nerve route, we have our preganglionic axon that leaves the spinal cord, goes out through the spinal nerve, and then what it does is it goes from the spinal nerve, or the anterior ramus of the spinal nerve, through the white communicating ramus, and then forms a synapse somewhere in here within the sympathetic chain of ganglia. That's going to give rise then to a postganglionic axon, and in this case the postganglionic axon goes back out through the gray communicating ramus and then merges back with that same spinal nerve that you were part of, oftentimes, and then goes on to its destination. So you're kind of like leaving the spinal nerve and then coming back to it, which seems kind of weird, but this is how it happens. And it does give, the spinal nerve itself doesn't have any place where you have cell bodies. This is basically the location of the cell bodies of the postganglionic axon. They're found here in the sympathetic chain of ganglia. So this is one way to do it, is you come out of the spinal nerve and then you go right back in through the white communicating ramus coming out and the gray communicating ramus going back in. Route number 2 is the sympathetic nerve route. And the difference here is we do not use the gray communicating ramus. So instead, what we do is it starts the same, lateral horn to the anterior ventral root of the spinal nerve, and then through the spinal nerve, and then you've got the anterior ramus, and then you go through the white communicating ramus. But this time, we'll form a synapse, and they're showing it kind of going up a level, and it often will do that. But this time you're forming a synapse that will then the postganglionic axon will not go back through the gray communicating ramus. The difference here is that instead of going back to the spinal nerve, we're going out this totally separate nerve that's called a sympathetic nerve. And that's going to carry our information until it finally hits your effector. So most of the signals go up to your head, also to your heart and lungs. They're traveling through these sympathetic nerves. And that's what it's saying here. This is the kind of destination that uses these sympathetic nerves. So those are pretty similar. The difference is basically where does the postganglionic axon go? In the first one, we went back through the gray ramus to the anterior ramus of the spinal nerve. In this one, we're just creating a whole new nerve, and we're going straight to our destination with our postganglionic axon. The final one is, I guess, the most different, but it's not that different. So this is called the splanchnic nerve route. So in this case, starts the same way, so lateral horn of gray matter. We go out through the anterior root, out through the spinal nerve, out through the white communicating ramus. But then the big difference here is that this preganglionic axon, which is shown in green, it does not form a synapse in the sympathetic chain of ganglia. It goes through it, but it just kind of bypasses it, like cuts right through and forms a new nerve. This is a totally new nerve, so it's not a sympathetic nerve because it's not. This is still the preganglionic axon, and it's going to a collateral ganglion somewhere. It's certainly not going back to the spinal nerve. So we call this thing a splanchnic nerve. So this is still the preganglionic axon that started way back in the spinal cord, but it goes out through the splanchnic nerve till it gets to some other ganglion, and that's where it's forming its synapse. So now it's synapsing with a postganglionic axon that can then continue to the destination of your choosing. So the big difference here is that that preganglionic axon did not form a synapse in the sympathetic chain. It just kept on going and didn't form a synapse till you finally get to this collateral ganglion way out here in the abdomen somewhere. And if you look at, there's sort of these two, it's like one image, but I broke it in half. So there's this one, and then I'm going to flip 2 slides. Then there's this one. In this case, the things you can see, I think the best on this one are the sympathetic nerve routes, where you have preganglionic axons, which are still shown in green here, that form a synapse in the sympathetic chain and then give rise to postganglionic axons that travel in sympathetic nerves back up into the head or to the heart or to the lungs. Down here, I think the thing this shows really well is you have these splanchnic nerves where a preganglionic axon will cut through the sympathetic chain of ganglia until it gets to some collateral ganglion. So that's the celiac ganglion or the superior mesenteric or the inferior mesenteric. And then at that point we finally form a synapse and give rise to a postganglionic axon that takes you to your destination. Yep. I mean, I think you should know that these digestive organs are getting serviced by splanchnic nerves because that's associated with these ganglia. I mean, I don't know. Like, how important is it? I don't know. I think it's fair game to know, especially these ones that we've explicitly talked about here. And it does kind of give you a list in these little red boxes. They're hard to see, but you can kill yourself over it, but it's fair game. I definitely think the collateral ganglia are distinctly different, and you should certainly know that most of your abdominal organs are serviced through these collateral ganglia using splanchnic nerves. I mean, to me that is a kind of a big point. And it does say, I think in text here. Well, I guess it just sort of mentions it again there. Both of them are hyperpolarized when they're stimulated by light. Yeah. I'm going to try it just here, and then if we need to go back into the rod or the cone. So the story is that rods and cones basically work in the same way. The only difference is that cones are less sensitive, and therefore they're able to be still active under these kinds of levels of light. Rods are so sensitive that they are completely overwhelmed at this level of light. So rods are only functional in pretty dark conditions, like if you're walking around with a half moon or lower, kind of illumination. That's it. Which would be really important if you're a caveman. In our kind of modern day, it's pretty rare that we really need to use our rods very much. But they work in the same basic way. So when they are not receiving light, they're in this sort of condition, and at that point you've got this thing called the dark current, which is where you have these sodium ions that are rushing in through these little channels. And that's happening all the time when these guys are not stimulated. So there's sodium ions, they're coming into the cell. That's going to cause the membrane to be fairly depolarized. They're still negative. It shows they're like at minus 40 here. So the membrane potentials are still negative, but they are less negative, so I guess more positive, than they will be later on when we actually have light encounter these things. As long as these rod cells are fairly depolarized and less negative, they will be releasing, you can think of them as kind of being excited under dark conditions. So what I mean by that is they are releasing neurotransmitter at this moment onto bipolar cells, and the neurotransmitter here delivers inhibitory postsynaptic potentials onto the bipolar cells. So in the darkness, the rod cells are fairly depolarized, but the consequence of that is they're releasing neurotransmitter that shuts down bipolar cells. And because the bipolar cells are shut down, they're not releasing neurotransmitters onto the ganglion cells, and therefore there's no signals being sent up into the brain telling you that there's light there, because there's no light. What happens then is that if there is a photon, or more than one photon, that hits this rod cell, it'll receive that photon and it'll cause rhodopsin to change its configuration. So retinal changes from cis to trans, and that's all the slide or two before this. The consequence of that is that it shuts down this channel, because you start to break apart the cyclic GMP. And if you shut down the channel, you stop the dark current, so there's no longer sodium ions rushing in. That means the membrane is going to become less positive and more negative. So the membrane in this case is shifting from minus 40 millivolts down to about minus 70 millivolts. So you are inhibiting the rod cell by exposing it to light. That's how it responds to light, is it gets inhibited. If it's inhibited, it's no longer releasing neurotransmitter. The bipolar cell is like, oh my God, this is wonderful because I'm no longer being inhibited by this rod cell under this condition. So it is disinhibited, and therefore it is free to start releasing its own neurotransmitter. And when it releases its neurotransmitter, the ganglion cell says, oh, okay, and starts sending action potentials up into the brain, telling your brain that there's light there. So again, like the big picture is you expose rod cells to light, and then that results in a change in the behavior of the rod cell and the bipolar cell. And the result is you get action potentials going up in your brain saying, hey, there's light there. The way it achieves that seems a little bit backwards. You're stopping inhibitory signals here, which allows you to start making excitatory signals here. But it still is, light's causing a change in memory potential, which changes the behavior of the cells, which then ultimately results in signals going up into your brain. I hope that helps. Like that, it is one of these things you just kind of have to stare at it. But again, like the big picture is we change the activity of the rod cell because it received light. And that's resulting in signals being sent to your brain. Rods versus cones, you mean? Rods would have more. The main reason, I think, if you look at a rod versus a cone, the major difference is the size of this outer segment. And this is where those little discs are. And the discs, so here's the discs. the discs are all studded with rhodopsin, which is this whole pink thing. The retinal plus the opsin is a rhodopsin. So because there's more space here, that means there's more discs in a rod, which means there's more rhodopsin in a rod. And that's why they're more sensitive. Whereas cones don't have nearly as many discs, so they have less rhodopsin, so they have less of a chance to capture light. And the result is that they will only really get excited under decent amounts of illumination like we have here. My mom just got a horse, and this is sort of a random story. Sort of a late life crisis, I guess. That she wanted to have a horse, and she didn't have a horse for so many years. So she just posted a thing that horses supposedly take about 30 seconds for their eyes to acclimate if they're going from outside to inside the barn. So they're basically blind when you first lead them in there. And so presumably what's happening there is that they were relying on cones out in the pasture, bring them into the barn, and their cones are no longer getting the amount of stimulation they used to. Their rods would have been totally swamped out, so you kind of have to let those readjust. I'm guessing they're still honestly using their cones as I think about it. But this is the basic idea between rods and cones, is that rods are useful under really low illumination levels because they're so sensitive, but they're blown away under high light. And so you need to give them time to acclimate and stuff once you get into darker conditions. And sort of undo that bleaching that they've experienced. Yep. In the retina. So they're in the photoreceptive layer. So when you look, it's a really, we didn't get a chance to talk about it, but the weird thing about the retina, again, it's one of these things, I think our retinas are bass-ackwards in so many different ways. That's as my dad always says it. In that the photoreceptors, if you look at the layers of cell, it's actually ganglions. The ganglia cells are at the top, and then you have the bipolar cells, and then the photoreceptors are buried like 3 cell layers deep into the retina. So light has to travel around the ganglia cells and around the bipolar cells to ever get down to those photoreceptors. And they're even upside down so that the photoreceptive part of the rods are the furthest possible distance from the surface of the retina. But they are down in there. That's just the way it evolved, though. If you look at an octopus eye, it's the exact opposite. And in some ways, they function a little better as a result. But, ours work pretty well. But that's why we have a blind spot, because all those ganglion cell axons, I'll stop talking in a sec, but these ganglion cell axons all have to converge on the surface of the retina and then go through the retina again because they're up on the surface, so they have to cut through the retina. So we have a spot where all those axons are going through the retina where we can't have photoreceptors there because it's all designed upside down. It's dumb. But it's because it's not intelligently designed. It's just that's how it evolved. So anyway. Yes. Yeah. So the fun thing about olfaction G protein system is that if you look at it, and I'm going to do this real quick, versus, if we go all the way back to chapter 12. We talked about how norepinephrine receptors work. And there they had a receptor that bound norepinephrine that activated a G protein that activated adenylate cyclase that made cyclic AnP that then had different effects depending on the cell you're looking at. And the cool thing is that these things work exactly the same way, except that instead of binding norepinephrine, they're binding some odorant. And every olfactory cell has a different odor receptor here. So they're, so you can bind different kinds of smells. But the idea is if the odorant happens to hit a receptor, that'll activate a G protein, which will activate adenylyl cyclase, which will create cyclic AnP, which will then go have effects. And in this case, it opens channels that causes these cells to start depolarizing. the same basic mechanism that we saw with norepinephrine receptor systems way back in chapter 12, which of course is also how norepinephrine receptors in the parasympathetic division, or I'm sorry, in the sympathetic division of the autonomic nervous system, they all work through that G protein system too. So it's the same sort of mechanism, and it's being co-opted and used in different ways. So the only thing that's sort of unique here, aside from I guess it's an odorant, is that in this case, cyclic AnP directly binds to this channel and causes it to open. But once that channel opens, it'll depolarize the bipolar cells here, and then they send their signals up into the olfactory bulb through your cribriform plate of your ethmoid. And at that point, then you're starting to sense smells. That's pretty much the story on them, though. It's sure. Yep. Okay, yeah, so the idea is that if something, so you divide your visual world in half, left side, right side, those are called hemifields, which basically means half of your visual field. All the information that comes from your left hemifield will end up on the right side of your brain, and it doesn't matter which eye it came in. So left hemifield information that gets to your right eye, that information, which is coded in yellow here, stays on that right side of your brain, and it never really has to cross over because it came in through the right eye. But if left hemifield information is captured by the left eye, that information has to cross over. So it goes up the optic nerve, and then that optic chiasm, which we always have to kind of wrestle with when we're doing the brain dissection because it kind of keeps the dura mater on and stuff. Those axons cross through the optic chiasm to get into the right optic tract, and then at that point they stay on that side of the brain. And then information that's from the same point in space, if it came in from your right eye, it'll end up in a cell that's literally like right next to a cell that got information from your left eye from that same spot in space. And then they can directly be compared. And that's how we start to get binocular, we use our binocular vision to measure depth perception and that kind of thing. But that's how it works in a basic sense. It's pretty cool. And, so like most of our vision, like you can kind of do this. And if you keep alternating between your left eye and your right eye, which one is open, which one's closed, like most of your visual field, you've got binocular overlap. So information is coming into both eyes for most of your visual field. It's only like when you get off into the sort of extreme parts of your peripheral vision that it's only coming in one eye or the other. So most of the time you have that situation where you have to kind of coordinate and get information from the left eye to merge over with the information from the right eye and vice versa. What else can we do? I didn't hear you, but I didn't hear you, but huh? No. No, I cannot give hints about content of exam. Yeah, I'm sure you do. It's definitely not a democracy, unfortunately. Like someday, you two will get to stand in front of a group, if you wish to do so, and become an authoritarian dictator. But for now, that's what I've worked toward, and so I'm going to claim that. Okay. Yes. Yep. Chapter 14, motor actions, how you make them. So we do a light version of that. Man, I swear I just changed. Didn't do it. There it goes. So we give kind of a glossy view of it, but this is the level I want you to know it for us. Let me get there. Yeah, we'll go here. Okay, so the frontal lobe is kind of divided into an anterior and a posterior part. It's all frontal lobe. So it's all the area that's kind of under your frontal bone. But this whole area, this prefrontal cortex, this is like, sometimes it's called the neocortex. This is the stuff that allows us to have personality and planning and conscious thought and all that jazz. So we're not worried about that. What we're interested in right now is the posterior frontal lobe, which includes all these sort of blue colored areas. I know it's kind of small, but you've got a lighter blue area here, and then you have a darker blue area that's right here. The lighter blue area over here, this is your motor association area. So at some point, I think your book calls it the premotor area. There's a couple of different names for it. This is kind of your, you could think of it as like the location where you generate plans of action, like motor plans, especially for complicated kinds of behaviors. And complicated behaviors, like the difference there would be the difference between just like grasping. Like grasping would be pretty simple sort of motor action. But a complicated behavior might be like reaching out and picking something up, or it might be playing a piano, or it might be, I don't know, any kind of like, well, certainly like writing is definitely, falling in this category of complicated motor actions. So the idea would be for that kind of an action that you probably make a decision somewhere in your prefrontal lobe about what you want to do. But the signals would then be sent into your motor association area up here, which will then cause you to kind of develop your motor plan. And this is, I think the other idea is that there's sort of like motor memories to be stored in this area. So you use those to generate a motor plan of what you want to do. And then you send that information to this structure in dark blue. That is your precentral gyrus, which is the very last, most posterior part of your frontal lobe. It's right in front of the central sulcus, so we call it the precentral gyrus. And its job is to actually send the signals down the spinal cord to the lower motor neurons that are in your spinal cord that will actually send the signals out then to the muscle cells. So the idea is that if you want to move your hand and do some sort of writing action, you'll develop a motor plan over in your motor association area. And then you send that signal into the hand control part of the precentral gyrus, which is the other word for that would be your primary motor cortex. And then it will get particular cells in these areas excited, and they will then send action potentials that will travel all the way down your spinal cord to the lower motor neurons in your spinal cord that will then send the signals out. So especially in terms of actions that are causing you to move your fingers, that'll go down the lateral corticospinal tracts, for example, which are the spinal tracts that control your distal muscles. And that will then ultimately result in the muscle contractions required to actually move your pencil on your paper. So again, the idea is you develop a motor plan, which in part might be influenced by motor memories or procedural memories in the motor association area or premotor area. And then you send that signal to the precentral gyrus, which is the primary motor cortex. And then that's what sends a signal actually down your spinal cord. And that's pretty much what we said about it. If you want more, you can take neural. We do a little bit more detail. A little bit more. Yeah, these more. Yep, sorry. Sure. Is that all good? Any questions on this before we leave? I just want to make sure. Okay. Cerebral spinal fluid. So if I wrote that question, I would be looking at this slide probably. So the idea with cerebrospinal fluid is we generate it with these structures that are called choroid plexuses, and we have them basically in all the large ventricles, but the first ones in series, more or less, would be the ones that are in the lateral ventricles, which are these two sort of side-by-side ventricles that go through both of your two cerebral hemispheres. So they're kind of overlapped. You can't really see the two of them, but they're like all this area. This is all lateral ventricle. And there are these little red spots here. These are choroid plexuses. So what they're doing is that they are basically, they're a special type of capillary bed with some other modifications. So they push blood through these structures. And the plasma part, the liquid part of the blood, gets filtered out into that and becomes that fluid. So the fluid itself is actually coming from your blood plasma after you've sort of filtered some stuff and added some stuff to it. So we start making cerebrospinal fluid in the lateral ventricle. It flows down into the third ventricle where there's another choroid plexus that adds to that cerebrospinal fluid. That fluid will then flow down the cerebral aqueduct to the fourth ventricle, which is down here in your brainstem. Where again, I think there's another choroid plexus that's down here. But then from there, you kind of have two options. One is you can, some small component of it will flow down the central canal of your spinal cord, so all the way to the end of the spinal cord. Most of it will actually exit the ventricle system and get out to the surface of the brain, where it will travel through the subarachnoid space, which is just like kind of like this small little gap underneath the arachnoid mater, so between the arachnoid mater and the pia mater. And it will then basically circulate and bathe all the exterior surface of the brain, as well as the exterior surface of the spinal cord once it gets out there. Eventually, it'll make its way up to these little arachnoid villi, which allow that fluid to exit from the subarachnoid space and get back into our venous system. So this is one of these big blood vessels, venous veins that are found up in there. We call them sinuses, but they're basically big floppy veins that are present all around our cranium. And they're receiving that cerebral spinal fluid finally. What else can we do? I can certainly go another 5. I can go more than that even, but. Or we can call it. I could. Probably. Yeah, so I can imagine functionally I could say like which of the following tracks does this, and I'll list a bunch of functions. Or I could say, and I'll try to make that really, I'll probably use the language on the answer key if I'm doing that, because there's like a couple that do pain, for example. So I could just say pain, or there'd be 2 right answers. But yeah, or it could read, where does the lateral corticospinal tract decussate? And you might have to indicate where it crosses over. What else might you have to know from that? Those are probably the main deals. Where is it coming from or going to in the brain? Be another one. So tectospinal pathways coming out of your tectum, which is basically your corpora quadramina. That kind of thing. Yeah, that is a thing. Sorry, I'll give you a second. I will say I've had, I think it's been better the last year or 2, but I had 1 year, I remember in particular, where like, I remember even standing there in lecture saying, make sure you do this and know these because they will show up in the exam. And then like nobody got any of them right, and it was clear nobody ever looked at them again. So, but you should they'll be on there probably somewhere. Yeah, sorry.
You said it's just like the main points from all the other chapters that are going to be on there?
Yeah, they'd be more like what I consider to be main points, or what I consider to be, the other thing would be if they're directly connecting and tying to the most recent batch of material. So like the stuff we did with norepinephrine receptors, just because we just did it, is a good example of like, so G protein systems. I guess you could debate whether or not that was a major point in the nervous system chapter, but we've definitely had it again, so you should know G protein systems pretty well. But again, that's always like what I consider to be, I don't know. It's not that I'm like trying to fool people, but sometimes people disagree with me about if something is a major point. But man, I'm trying to not find some obscure term from chapter 6 and play getcha. It's not my goal. But yeah, go ahead.
So if you're asking this for like nerves and really different muscles,
Yeah.
just give us like the different muscles for like the language and something.
I would probably be looking, when I'm writing that, I would think I'd be looking at the answer key I gave you for that stuff. So, this one, right? So that's mostly this stuff where, so these are all these nerves that come out of the different plexuses. So, you know, over here with musculocutaneous nerve, you know, I'm sort of listing these muscles. So I'd probably guess, if I was going to write that question right now, I'd probably write it with explicit, like using that language probably, or something really similar to it. I'm also noting that I didn't close my parentheses, and that's really bothering me right now. But I'm in a PDF, and I have to go back to the original. But that I think would be the idea. So I don't think you should spend, unless you find it helpful and easier to interpret, I don't think you should spend a ton of time with the muscle list, because I know it lists innervations, but what's on that muscle list should match up to what's listed here. So if you're learning them in groups and you know basically where your muscles are, you should be fine. There won't be a ton of those, but you know, I could see there'd be a couple questions like that. Anything else? Once, twice. Sold. All right. Well, best of luck. I will see you tomorrow at 10:10. Good luck studying. I will make sure I check my email